Hello, in this video we will discuss about the Sanger method, Sanger sequence of the DNA. So what is that? That is very important topic we will discuss, and that is regard to the examination, etc. So here is the Sanger sequencing developed by basically the Frederick Sanger and colleagues at University of Cambridge in 1977, and this involved in basically involved this involved in vitro DNA synthesis. So in vitro DNA synthesis is very important, and in this way we will discuss whole the mechanism. So the base of the principle and biochemistry of DNA replication, and this is the base on the principle and biochemistry of DNA replication and Sanger DNA sequencing. So let's begin to understand the requirement in vitro DNA synthesis. So the DNA template strand should be available for that. So the DNA template strand. On the other hand, the complementary sequence of the primer should be. So that is the complementary primer should be which that is used for the annealing process from 5 prime direction to 3 prime direction in the anti-parallel. So this is the DNA template strand. In this way, the DNA template strand is 3 prime to 5 prime. So the 5 prime to 3 prime annealing process for the primer will bind. If the primer will not bind, so the annealing process is impossible because the DNA synthesis will be impossible. We will also understand why it is impossible. And on the other hand, the primer serves the starting point of a new strand synthesis. So the next thing is that the deoxynucleotide is very important. DNTP DNTPs is the basically is contain deoxynucleotide adenine triphosphate and cytosine triphosphate, guanine triphosphate, as well as thymine triphosphate. So these four type of deoxynucleotide is required for the synthesis of DNA. Let's begin to understand. Please concentrate on this diagram for understanding. So here is the 5 prime direction to 3 prime direction extension will be occurred through polymerase enzyme. So the DNA polymerase is also required for this. And in this way, the nucleotide will be bind from 5 prime to 3 prime direction anti-parallel from the DNA template strand. So the primer is essential for DNA synthesis. Why? Let's begin to understand why primer is very important. So the polymerase cannot synthesize DNA from the beginning its own. So primer is very important for begin the reaction. So the beginning of the reaction is very important. Why? Because the polymerase cannot synthesize DNA from the beginning its own. Because here I am drawing the DNA structure, you can understand as well. So this is the phosphodiester bond and this is a hydroxyl group. And in this way here is the nitrogenous base and deoxyribose sugar. And here the primer provides the initial 3 prime hydroxyl group here. And this is suppose this is the primer and group to form phosphodiester bond with the incoming deoxynucleotide triphosphate. So in this way that is very important because the primer contain hydroxyl group so it can be bind, and when DNA grow the 3 prime side contain hydroxyl group, so the group and thus polymerase keep grow elongate the DNA chain. So that is the very important things. Otherwise if you think about that the nucleotides, so the nucleotide is the basically single which that is contain uh, a nucleotide is not a property for that because it will not run from 5 prime to 3 prime direction, and that is why the one by one nucleotide can be added in the front of the primer to elongate from the annealing process. So the based on the principle and biochemistry of DNA replication, and this is the basic for the principle and biochemistry is very important. And basically, the Sanger DNA sequencing technique makes a use of modified deoxynucleotide known as dideoxynucleotide. It means the hydroxy group here is the deoxynucleotide, which that is here is you can see adenine, thymine, guanine, and cytosine are attached the base and triphosphate. So this is the nucleotide. So the here is the dideoxynucleotide because the carbon number 2 prime and 3 prime which contain a only hydrogen, so that is why oxygen are not present, so we can say that dideoxy because two oxygen lack from the carbon number 2 and carbon number 3. So here the deoxy nucleotide triphosphate are also known as the chain terminating nucleotides, and because the carbon number 3 lack oxygen, so the Sanger method also known as the chain termination method or dideoxy DNA sequencing. So that is why it is very important. This dideoxynucleotide is used for the termination with a different region. So in this way, here, let's begin to understand the Sanger sequencing. So the common reaction component is DNA primer, DNA primer, many copies, radio labeled, and this is the radio labeled. And in this way, the radio labeled primer, which that is will show the amplification of the gene and show the color, and we can say that this color is the segment of DNA which that is after the synthesis. So the DNA polymerase is also important as well as the nucleotide DNTPS, and that is the DNTTFPS and DATPS, DCTPS as well as others. So the large amount of these nucleotides should be. Why? Because the synthesis of the DNA required this nucleotide, so that is why. But in this case, we are adding the dideoxynucleotide. Each reaction contains separately contain one deoxynucleotide. For example, here is the DDATP and DDCTP and DDGTP and DDTTP. So in this way, here is the polymerase enzyme will be bind, and the nucleotide will be bind. After this nucleotide will be bind, the deoxynucleotide is the one type from each reaction contain one type out of four. So in this way, the basically the termination will be occur with the specific point where the complementary strand will be synthesis. I am making this reaction with a briefly. You can understand as well. Here is the here is the deoxyadenine triphosphate. If we will put, so this in this way the reaction we can be inhibit or terminate from this side, T side, so the thymine side, because the adenine always bind with the thymine. So the terminal point, why? Because the oxygen hydroxyl group is not present in the deoxy 80 adenosine adenine triphosphate. So in this way, here is that there are different segment will be produced. And similarly here you can see deoxy cytosine triphosphate if we will put that. So in this way the guanine will be opposite anti-parallel strand will inhibit. So there are the three strand can be developed after the termination with a different region. For example, G and GG. So on the other hand, similarly here you can see the deoxy GT guanine triphosphate. So in this way, here is the basically the two segment can be occur after the termination with a different region. For example, the cytosine. So in similarly here you can see the deoxy thymine triphosphate, which that is produced the four type of the segments. So this all segment we can collect and we can put in the well of the gel electrophoresis, polyacrylamide gel electrophoresis. When we will put these things, adenine, cytosine, guanine with thymine 1, 2, 3, 4. So on the basis of the density of the segment of the DNA which that is synthesized now will be separate from negative to positive charge after the providing of the electricity. So here we can calculate through autoradiograph obtain radiation will produce color, and in this way here is the sequence we can calculate here, and we can put this G T S G A T C C C G A T A C and T on the basis of downward to upward, low density to high. So in this way the large fragment will remain the close to the negative charge while the small segment will move forward, and in this way we can say arrange in this manner, and to synthesize, or we can say this is the antiparallel strand is original template strand. So this is the anti-parallel strand was the original template strand, which that is we are getting that things. So this is the important things. It is also used for the fingerprinting, and it is also used for the identification of DNA and DNA products of the different region but same starting point. So that is important thing, and where primer will be bind but the termination will occur in different region. Primer will bind with the same region, but the termination will occur on the different region, on the base of different region. So you can see and understand why, because the d-deoxy nucleotide will bind when, so the termination can be occur, otherwise will not occur. When deoxynucleotide will bind, so the elongation will remain constant, but when the deoxynucleotide will be bind, so the termination will be occur to produce several type of segments. Thanks.